Hi, my name is Orly, and our group is going to be talking about associative tolerance to drugs. Here are some keywords and definitions.
Hi, this is Sophie. So the definition of associative tolerance to drugs is tolerance to drugs that develops in the presence of predictive environmental cues.
Hello, my name is Diane, and I will briefly discuss global context. Why is it important to learn and discuss associative tolerance to drugs? Drug use and associative tolerance to drugs affects many people globally. Pictured on the slide on the left is a global map depicting alcohol and drug use disorders as a share of total disease burden. The map measures disease burden in DALYs. One DALY equals one lost year of healthy life. This is important because substance abuse can result in an abundance of negative health outcomes other than death, including emotional distress of family members, economic hardship, injury, criminal records, and houselessness. All of these statistics point to the fact that we really need to focus on rehabilitation and effective treatment for drug users because many drug users do tend to relapse or return to their old habits. So, understanding addiction and its effect on the brain and behavior is really essential for creating programs that will prevent relapses. And we know associative tolerance is especially dangerous in that it can lead to overdoses, especially when the drug is taken in unfamiliar contexts. The first study that we looked at is the limbic activation during cue-induced cocaine craving. And so the study demonstrated that cues related to cocaine use trigger cocaine craving, and it did so by using positron emission tomography to look at the limbic systems of former cocaine addicts and subjects who had never used cocaine before. And these subjects were exposed to non-drug related videos as well as drug related videos. And so when videos contained cues that were common to cocaine use, it led to craving, and this led to an increase in limbic activation during these videos. So what this showed was that the cues themselves can be linked to signs and symptoms of cocaine, and it can also lead to changes in the brain, which can lead to cravings and cause relapses. And so this showed that associative tolerance exists in that seeing these cues can trigger these responses even if the drug isn't being administered. One benefit of the study is that it showed that associative tolerance exists and it showed the importance of the cues. Some limitations were that the video might have led to other responses, but there's no evidence to show that this occurred. And there might also be some difference between cocaine users and non-cocaine users, but because participants were screened, there were no clear differences that were found that could be causing this. The second study looked at preventing the recovery of extinguished alcohol tolerance, and this used a factorial design to look at the importance of context and also delay between when the drug was administered and when extinction occurred. And so basically, the study found that mice had developed associative tolerance, and this tolerance was effectively extinguished as a result of extinction trials. And it was also found that inducing a delay between the extinction and the reacquisition led to spontaneous recovery, and testing outside of the familiar context led to renewal. And so there was an interaction between these factors indicating that testing outside of the extinction context led to more recovery of tolerance, and there was a short delay versus a long delay. And one great benefit of this is that it shows how important context and time is when it comes to preventing relapse. And some possible limitations were ceiling effects that prevented further effects from being seen in the data. And one great future application could be using this to inform exposure therapy.
In this experiment, they were interested in testing if unsignaled morphine delivery has an impact on associative tolerance, so they conducted 3 experiments. And some of the things they did for the procedure is they used 123 male husband rats, and they kept them separately in a cage in a colony room. There were 4 treatment conditions that the rats were assigned to. First condition consisted of morphine, prime distinctive context. The second condition was a distinctive context. And the third condition was the home cage. And the last condition was the saline control. And this is a tail flick device that they used. For the graph, this shows the mean tail flick latency 30 days after last conditioning session for each dose group as a function of morphine dose for each condition. A conclusion of the study, after conducting all 3 experiments, they did not find that pre-exposure to morphine in short interdose interval disrupts are responsible for disruptions in the acquisition of associative tolerance effects when testing rats immediately after conditioning. Their study indicated self-generated priming of morphine is not powerful enough to cause a disruption of the development of associative morphine tolerance. So analytical thinking, future studies choose different drugs to see what the results are with those drugs. They use a 6-hour interdose interval, which may have been too long, in addition to the priming dose, which may have been too low. They also did experiment on male rats, which may have affected the results due to their biological makeup. So these are some weaknesses, and some strengths are they conducted 3 experiments to fully test the hypothesis. They also used the same type of rats and kept the rats separately in the cage, which are important controls. The morphine was dissolved in saline to ensure the dose was isotonic with physiological saline. In this study, they were interested in testing the effects of different amounts of conditioning trials impact the development of associative tolerance to drugs, and they gave the rats different administrations of the drugs. They used a tail flick device. They did about 3 consecutive trials while the location of the light varied. For the tolerance development phase, there were 3 conditions. The first condition was a distinctive context, the second condition was the home cage, and the last condition was the saline control. And so here are different graphs of the different treatment groups and the condition groups. And so overall, these graphs showed that the rats that were in distinctive context and had morphine paired experienced the greatest morphine tolerance, and in the condition where the rats had morphine not paired with a distinctive context, they exhibited less tolerance to morphine. In conclusion, rats plus morphine plus distinctive context equals more tolerance to morphine, and in the case where the rats and the morphine and no distinctive context, there were less tolerance. So analytical thinking, this implies that many times when it comes to the drugs, the environment play a huge role, as we can see from this article. Many times there are environmental cues that are associated with the drug. Strength in this experiment were that they used a large sample of rats with increased accuracy. They kept the rats individually housed, and they used the same type of Halsman rats, which were controls. Weaknesses in the experiment were that they did not test rats for any biological predispositions, which may have impacted the effects of the drug. They used Halsman rats and only tested associative tolerance to morphine on the same type of rats dose. Rats does not show its effect on larger scale. It could be that these rats were less tolerant compared to other rats, and additionally, these rats were all the same age.
This is Diana. Here I will discuss an article published in 1997 by Cox and Tiffany. The study conducted 2 experiments to investigate the effects of dose and interdose on associative and non-associative tolerance to morphine analgesia in rats. The first experiment examined the effect of environmental contingencies on tolerance development when high and low doses of morphine are paired or unpaired with distinctive environmental context. The second experiment deployed a 30-day retention test to compare tolerance developed in the low and high groups. Here are the major findings of the article. The following images and captions are taken directly from the research article. The images depict the mean tail flick latency on the test sessions for each dose group as a function of log morphine dose for each of the treatment conditions. Beginning research on associative tolerance to drugs, including Siegel's theory, fails to provide an explanation as to how tolerance can develop in the presence of drug-predicting cues. Later research, including research published by Tiffany in 1992 and the Baker and Tiffany habituation model, examined tolerance as a process that contains associative and non-associative characteristics. A major strength of this paper is that the findings are consistent with those of Tiffany et al. In 1992 and offer support for the Baker and Tiffany habituation model of drug tolerance. By expanding the way that tolerance is examined, researchers in this study and the ones that inspired this research are able to garner a more thorough understanding of how tolerance can develop within different environments, which is important for preventing overdose. However, this study also has many weaknesses, including having no knowledge of the mechanisms responsible for certain effects, such as the disruption of sure IDIs. Future research can be directed towards identifying such unknown mechanisms. The next study I will be discussing was published in 2000 by Carter and team. This study investigates the associative and non-associative tolerance to the analgesic effects of fentanyl in rats. The methods used in the study are depicted below. Findings of this study include the discovery that contextual contingencies had no effect on tolerance of the subjects. This finding suggests tolerance was controlled by non-associative mechanisms. The 3 following slides include images and captions taken directly from the study. The graphs depict results for animals conditioned using non-associative tolerance procedures with tail flick latency on test sessions as a function of test drug log dose for each treatment group. The table depicts the average dose producing the half maximal effect on the tail flick for various substances throughout various contexts. The major finding of this study is that associative fentanyl tolerance is cross-tolerant, which suggests there is a conflicting biological process that supports associative tolerance in the mu receptor. A major strength of this article is the discovery that findings of non-associative morphine tolerance are not necessarily applicable to other opiates, including fentanyl. A weakness of the article is that there was not a concrete mechanism that explains the difference between fentanyl and morphine tolerance identified in the research. Future research can work to identify the mechanism responsible as well as identifying further differences between the opiates.
Hello, this is Sophie, and the purpose of this study was to measure the associative and behavioral tolerance to the analgesic effect of nicotine, and theories of drug tolerance differentiate between associative and behavioral drug tolerance. However, little research has been done previously on the effects beyond alcohol and morphine, and to test this, they used rats and tail flick procedures, which are spoken about previously and can be seen in the image on the left of the screen. And the study tested varying time course development to measure the effects. And the rats were broken into groups of 1, 5, 15, 10, or 20 and administered that many doses of nicotine, either explicitly paired or unpaired with a distinctive context. And the study found that the associative tolerance developed quicker when nicotine and the tail flick context were paired versus when they were unpaired. And one major problem with the study is the amount of nicotine is not explicitly stated in the method section, and this information would be necessary for someone to repeat the experiment. And a strength is that it agrees with previous data and the initial hypothesis, and the previous data is based off of alcohol and morphine. And then here is the results section, where you can see the tables of the results of the tail flick, as long as the analytical or numerical data. In terms of analytical thinking, the American Addiction Center has stated the most addictive drugs are cocaine, heroin, alcohol, nicotine, and meth. And so greater research done on those drugs and the relationship between associative tolerance would be useful in further understanding how addiction works and the relationship between addictive drugs and distinctive contexts and associative tolerance. And this study found that the tolerance to morphine in rats associative non-associative effect by Tiffany and Maud Griffin found that the only rats that received morphine paired with a distinctive context were tolerant to morphine. The magnitude of the associative tolerance was a positive function on the level of the conditioning dose. And this can be seen with the graph to the right. Additionally, the weaknesses of this experiment are that they only use male rats, which could have affected the data. And some of the strengths are that they use the same type of rats with the same life experiences. And one reason that this is useful is that we can use this information for studying recreational drugs in the future.
In this article, we could see that learning predates life and actually begins prenatally. This makes offspring vulnerable to early onset of addictive behaviors maternally. Early exposure to alcohol through lactation were more inclined to exhibit addictive behaviors through maturation, and we could see this in the graph. For the methods, the creation of episodic pattern of excessive alcohol drinking during pregnancy and lactation in between group design with pregnant mice and evaluation of offspring mice for cocaine-induced condition place preference. And we could see these different graphs. And for the results, how would the behavior of female rats be impacted by prenatal alcohol exposure? Alcohol was evaluated for its sensitizing of cocaine-seek behaviors. How would prenatal exposure to cocaine influence cocaine-seeking behaviors? How would prenatal exposure to alcohol influence alcohol-seeking behaviors and alcoholism? So these are some questions that we have. Some strengths are neurocircuitry explanations. Weaknesses are explanations of significance between results of mice with different mutations and how we would see other behaviors comprised. Only male rats were included in the study, which is of weakness.
GABAergic systems and addiction share an association in both mice and humans. Through the mechanism in humans, it's unknown. Mice develop addictive behavior significantly influenced by alpha-2 GABA receptors and dopaminergic pathways, causing their vulnerability to triggering cues for relapse and behavioral sensitization to develop tolerance. The methods use genetically manipulated mice to evaluate how receptors influence locomotive activities, electrophysiology, and behavioral responses. And limitations for future studies and analysis is there's a feedback loop between the tolerance and compromised locomotive function, and does this relationship increase risk to relapse? And how can we distinguish altered expressions from lack of expression and determine correlation between gene coding and addiction behavior? The strengths and weaknesses are below, and the results are to the right.
So extending these different experiments to the global context of why this is so important. So we know that we're looking at associative tolerance to help get a deeper understanding of the current drug crisis. And so studies such as those by Gonzalez help us to learn about how we can prevent and extinguish alcohol tolerance. And this is really important for developing effective treatment. And experiments such as the Beta Bonitos demonstrate that context is really important. So this also provides insight into addiction and how important context is when it comes to associative tolerance. And so this knowledge can now be applied to possible treatments or therapies. And it has also been found through studies such as Carter's that different drugs require different models of understanding. So there's still a lot of work to be done to fully understand the mechanisms of drug use and addiction. And so some further research should be done on various types of drugs to study their unique effects on associative tolerance.
In articles by Mogul and Bailey and Dunn in 2012, they explain how gender plays a role when it comes to drug tolerance, and research has shown that women are less tolerant than males in terms of drugs. So based off of this, we wanted to extend this previous research and ask, does gender impact one's tolerance to drugs? So looking at these studies, studies that have been done on drug tolerance, they incorporate more male rats. However, it could be that male rats are more tolerant than female rats. So therefore, more experience could be done investigating the realms of gender influences on associative tolerance to drugs. Additionally, we were wondering if the weather impacts drug tolerance. So based on an article by Hajat in 2010, it talks about the impact of hot weather on drugs, and it emphasizes how drugs can affect one's ability to survive in certain climates. In hot climates, drugs can impair one's ability in recognizing how hot it is and impact homeostasis, which is important for survival. So research to investigate if weather impacts associative tolerance to drugs, and further investigate if drug problems are more common in colder regions compared to warmer regions. And here are our references and our credits.